All right. It looks like we are ready to go. So welcome everyone, and thank you for joining us for Lab Central's webinar in the series Innovation and Biotech in the Time of COVID-19. So my name is Ries. I'm Vice President and Head of our Oncology Therapeutic Research Center in Cambridge, Massachusetts. I'm really excited to be joined here today by a panel of outstanding industry leaders to have a conversation around oncology and kind of what's happening in the world today. Just in terms of some logistics, we will have a 30-minute discussion followed by a 15-minute Q&A. And so during the conversation, we invite you to ask myself or our panelists a question using the Slido link, and we will be able to get to those questions at the end of the meeting. So without further ado, I'm going to introduce our esteemed panelists, and so I'll sort of go around clockwise. So Wendy, thanks for joining us today. Perhaps you just would like to give the audience a brief introduction about yourself and your background.
Sure, thanks, Emma. Hi, I'm Wendy Winckler. I'm the CSO of Glympse Bio. I've spent my career focused on using cutting-edge technologies to better understand cancer. I'm starting as an investigator with the Cancer Genome Atlas at the Broad Institute, eventually moving to Novartis, where I established and led the Next Generation Diagnostics Group, where we applied laboratory and computational methods like liquid biopsy, next-generation sequencing to identify biomarkers of drug response. And I joined Glympse about a year ago because I saw great potential for this technology to be the next evolution in oncology biomarker science.
Great, thank you, Wendy, and welcome. Rick, do you want to introduce yourself?
Thanks, Emma. So my name is Rick Gregory. I'm a translational medicine lead at Takeda. Prior to this, I spent greater than 13 years at Genzyme and then Sanofi when it was acquired, where I led a drug discovery lab there, working on multiple different programs in both oncology as well as fibrosis. I also helped there to really help establish our IO portfolio at Sanofi. At that point, I decided to really think about moving into more human clinical trials. So about 2 years ago, I moved to Takeda, where I started working in our early IO portfolio, our early IO portfolio from the translational medicine side. So here I lead a number of different IO aspects, including both internal programs as well as ones that are partnered with external companies.
Great, thanks, Rick. I would look forward to getting your input today in this discussion. So Stefan, do you want to introduce yourself?
Yeah, thanks, Emma. So my name is Stefan Ries. I'm heading the External Innovation Organization at Roche, Pharma Research and Early Development. So this team is a global organization. We are spread across 3 different Roche innovation centers, namely Basel, Zurich, and Munich. And we consist of a team of senior scientists and experts in oncology research, but also medical oncologists. And we work very closely hand in hand with our business development organization. It's called Pharma Partnering, in principle to identify, assess, and license or acquire high quality assets, or also technologies to complement our internal oncology pipeline. But we also landscape strategic fields where we are not yet invested to really identify promising areas early on, not to miss an important field. For example, microbiome, radiotherapy, gene therapy. These are all areas which could evolve pretty fast. And yes, for us, it's important to really not separate our oncology pipeline into an internal one and an external one, but to have one collective pipeline. So I'm really looking forward to this discussion today.
Wonderful. Thank you, Stefan. And then I did sort of briefly mention myself, but just so I work for Bristol Myers Squibb. I lead one of our thematic research centers based in Cambridge and Boston, focused on sort of early discovery and translational research, focusing on trying to understand and come up with new treatments for patients that are resistant to current therapies. So whereas Stefan is sort of external innovation, maybe I'm internal innovation for a large company. So again, I think we sort of bring some different perspectives to the table. So to kick off the conversation, I thought that given the topic in terms of sort of innovation and biotech in the current era of the sort of COVID-19 pandemic, I thought I'd launch with sort of perhaps asking the panelists in terms of obviously we're all here on Zoom rather than around a table, and which just represents one of the many adaptations we've had to make over the last few months as we've dealt with the pandemic. But perhaps you could sort of just share with the audience how the pandemic has sort of shaped how you go about your business. And Rick, I'm going to start with you because obviously you're sort of right at the front line in terms of working with patients and trying to get clinical trials up and running. So perhaps you can tell us a little bit about kind of that experience and how you've sort of overcome some of the challenges with the ongoing pandemic.
So thanks, Em. I think it's a great point. I mean, other than me being surrounded by my son's stuffed animals here, I think from a Takeda perspective, very early on during the pandemic, and when COVID-19 was very early being discovered, in early March. I mean, I think it was Takeda shut down pretty early, and we started working from home pretty early. We were fortunate in that one of our vice presidents of our vaccine division, Rajiv Venkarayev, he was actually under George Bush as sort of the pandemic chief and sort of helped craft some of this policy. So, so Takeda very quickly shut down. So obviously this was pretty devastating to us because, early on we did shut down, and that included some of our clinical trials. And the trial that I'm leading, was scheduled to have its first patient in April, and it's one of the highlighting programs within Takeda. But Takeda leadership got together as a whole and said, we're not going to put our patients through, potentially coming in and getting exposed to COVID-19. They took a very logical approach to this, and it's really been over the last month and a half or so that we've started re-engaging with our clinical sites to reopen up. And I mean, this was controversial for a lot of us because some of our sites and those that were in New York City, were very understanding and said, well, we probably couldn't even take a clinical trial at this point anyway, because so many of our clinical sites were taking their oncology floors and converting them into COVID spaces. So some of the sites, and then we had other sites that were actually quite honestly quite angry at us. I was going to use a different word, but they were quite angry at us because these early clinical trials offer these patients a last hope in many of these cases. So so it was tough for us in the team because we didn't really fully know what was the best option. I think most of our investigators knew, and we were very grateful for our senior leadership to say, this is going to be the Takeda stance, and it aligns with Takeda's what we call Takeda-isms, which are, to put the patient, to put our patients first. So in our best effort, we thought this was the best route forward, and we are starting to reopen our clinical trials, but we are doing it in a very slow and methodical manner, and we are taking a very good look at what's happening in those locales. One of our sites is in Dallas, where obviously we're all aware that in Texas, COVID cases are going through the roof. So maybe our first patient is going to be in Denver, where COVID is a little bit more under control. So that seems to be the approach that we're taking. So certainly, that's it's been a hard decision, and it's been hard on the teams because we're excited to move these drugs into the clinic, but we understand the pandemic going on.
No, thanks for sharing that perspective. And I assume, like for us at BMS, that you guys have been part of these sort of, across industry kind of conversations about what the right thing to do is. I mean, I think that's one of the things that I've been really impressed with, how the industry has come together and galvanized through this in terms of, helping each other think through best practices.
Right.
Stefan, so what's the land of BD like when you're in lockdown and can't interact, which is your business?
Yeah, thanks, Emma. Have to say in the past, we have identified many of, let's say, the interesting opportunities by having direct access to, for example, academic thought leaders, scientists, clinicians. Many times these were the typical international key scientific meetings like AACR, ASCO, ESMO, others. Also some business meetings. For us important, for example, is always the JP Morgan Healthcare Conference. And in this respect, we were lucky because, you know, in January, we still could have this meeting. And so all of those meetings, they are now not existing anymore, at least not in the form we know them. And many of those, you know, discussions that are really important, that trigger any meaningful actions also for us, they are not really the ones within the talks, within the presentations, they are on the sidewalks, on the floors. So in principle, these conversations we are missing at the moment. And the other thing is our business. I think our external innovation business also depends very much on a personal relationship building. Mm-hmm. So you want to know the partner, you want to talk to the people you want to work with in the future. And all this is, of course, at the moment not possible. We cannot have face-to-face meetings, or at least only very limited ones. We cannot do any visits. We cannot do any on-site due diligences. So overall, I think this is quite impacting us, but I have to say, as I said, many of these meetings have become virtual. So this is really a good thing. Also, it can not fully compensate maybe for the many personal interactions and the much more extensive information exchange. But I think we are still pretty well informed about novel developments in our field. We have established relationships. We have access to literature, competitive intelligence. So at least we know what's going on in the field. And I think we are also now learning how to move discussions with potential partners forward in a virtual format. Mm-hmm. So for me, it's a lot of learning that we can actually achieve a lot by virtual meetings, and maybe even traveling like we did in the past. Right. You needed to you jump onto a plane and fly to the US and then have a meeting and fly back. I mean, all these maybe not so good things of these visits, like jet lag, you are exhausted when you arrive. This is at the moment not existent. So this is the good thing. The other thing I want to mention, maybe, which is also important for us, we have like a requirement at Roche that we at some point decided to do is we always want to reproduce some key experiments for an external asset before we license it, at least when it's a preclinical asset. If it's in the clinic and there are already clinical data, maybe this is of less relevance. And at the moment, as we heard before, our labs were also shut down. So we couldn't run all the MTA-based experiments, and we had to go to CROs outside in countries, in places which were not so much affected. So this delayed sometimes a little bit the process. But I have to say, because overall everybody was affected to some extent, right, globally. Overall, I think we could see a lot of understanding in our partners, and I think we could handle it. And you see that in the first, I think, 2 quarters of 2020, there are so many deals done, high impact deals have been done. So it seems like there's not a lot of immediate negative impact.
Yeah, no, thanks, Stefan. No, I think you bring up a lot of good points. So Wendy, so you're in a sort of pre-IPO small biotech with a sort of limited runway and finances and things like that. Obviously the bigger companies kind of have a bit more sort of solid financial, that they can sort of absorb some of this. So what's it look like from a small biotech that is kind of, where timelines are so critical? How have you sort of dealt with this? And, I know that, congratulations on just closing your Series B, but perhaps you can tell me a little bit about sort of life in a biotech and the issues that you guys have had to face.
Sure. So it's definitely been challenging, although I think we've been quite fortunate. Like most places, we did largely shut down operations back in March, but we did keep our essential lab projects that were IDE enabling going throughout the period. We had arranged a tool such that we could have a maximum of 1 person in the facility at any given time, and tracking that very carefully. Everyone was incredible. Like the lab staff was incredible about their diligence and coming in, following directions. We actually had a COVID exposure not within our lab, but in the wider building that we then had to do full contact tracing for, and that all went quite smoothly. And we didn't actually skip a beat and through all of this. And we manufacture our drug supply in China. That was going on in April, and yet that managed to actually stay on track despite all of the issues they were having there over the spring. So we were able to keep things moving, both at our CROs and internally. I would second a lot of what Stefan's been saying about the travel. Trying to close, a financing round normally would have involved a tremendous amount of travel. We do a lot of business development discussions. It's been amazing to me how much of that has been possible to do virtually. And I think it's going to actually change a bit about how these things are done in the future.
Yeah, no, great. I think that's a great segue into sort of the next question, and perhaps I'll go to Stefan first. So rather than have a long conversation on this, so I think we're all sort of hoping for kind of return to normal, but kind of thinking about what the new normal is going to look like, and kind of what have we learned through this experience when we talked about sort of jumping on planes and doing all the crazy stuff that we were doing that sort of felt like part of that you had to, and we've sort of realized that you can do things in many different ways that we wouldn't have anticipated. If you can think about sort of one thing that you're going to kind of do differently in the new norm that has kind of been a lesson learned for the better, through this pandemic, perhaps you could sort of share that with the audience. So Stefan, I pass that baton to you first.
So I think for me, the key learning and the key experience was that the technology nowadays, maybe which I was not even aware of, like all these virtual meeting tools, they have so much advanced that at least for existing relationships, it may not be always needed to really meet face to face, because the people, you have already established some trust. You can do many things maybe by a virtual meeting. And what I also learned, when you have a virtual meeting where everybody is connected individually, sometimes the quality of the meetings is higher than if you have 10 people in a room and 3 are connected virtually, because then the discussion often is still in the room, and the 3, let's say, that are only connected virtually are not, in the room, so they are less contributing. So for me, this is the key learning. We can do a lot with virtual interactions, and in particular in established relationships.
Yeah, no, I mean, I could second that in terms of for these big meetings where you have lots of different people calling in and, and asking questions during the course of the meeting can really throw off a meeting, right? And then meetings, run over. Now being able to use the chat function, you can have a really, great conversation going on. Everyone can contribute, everyone can sort of see the course of the conversation, and yet you can still have the presentation ongoing. So I think, that'd be something that, why have a meeting when you're trying to cram everyone in a room when you can have a much better quality conversation using one of these new platforms? I couldn't agree more. Rick, how about you?
I think it's, I think I concur with Stefan. I think I was actually really impressed with how quickly we switched over to virtual meetings and and moving to this. I think part of it was really about training myself to to be able to do these, because I always had I had a lab. I'm an in-person type of person, that if I had to go talk to somebody, instead of picking up the phone, I would walk over across the street to the building and just try to have that personal conversations. So I think for me, it is about learning how to stay to who I am, to pick up, we use Microsoft Teams a lot, to be able to just, call up somebody and just say, let's have a conversation. So I think it's, part of it's about, using the technology. Part of it's also about training yourself to, to use the technology and, not, adapt to it. So I think, for me, it really was about, just, getting used to it. When do you use WebEx? When do you use Teams? When do you use Zoom? So, it is a lot about that. So I think, I agree. And it is amazing what you can get done. And, and also still, challenge. I have a 4-year-old son. So balancing, this, really was a challenge. And I think you learned a little bit about yourself too.
And you got to know his stuffed animals better.
Yes.
Wendy, how about you?
Sure. I think one thing that we're realizing is that this, when COVID-19 is over, that this may fundamentally change how we work in terms of the need to be present in office all the time. So I think we have some people who have adapted to working from home brilliantly, love it, find themselves to be more efficient. Some of us prefer to be in the office a little bit, and some really like a blend. Like I'm in the office today. I'm grateful that now that we're a little bit reopening in Cambridge, we're able to do more of that. Our lab team has to be in. So we're naturally going to have some sort of a blend, but I think that flexibility will allow for different work styles and also different stages of your personal life.
Yeah, I mean, I think it's going to be interesting. We have not, sent our office-based people back to work yet. Again, we don't want to, increase. We're trying to get our lab people back. But yeah, I mean, I asked the same question, of myself. It's like, when am I going to go back in? And for what purpose? What, what's that new norm going to look like? So it's going to be interesting. But thank you, everyone, for sort of sharing those insights and kind of how the pandemic has sort of influenced what's going on. Let's kind of get into, a little bit of in terms of kind of, what are we thinking in terms of oncology, since we're, pushing and innovating in different areas. And, let's see, who should I jump to first? Maybe I'll keep on Wendy, since you're there, Wendy. And so perhaps you can sort of tell me a little bit in terms of, again, as you're getting up and developing, Glympse, what does innovation mean to you, and kind of where, what are the areas of oncology that you think are sort of really novel and up and coming right now? What excites you?
Sure. So let me tell you a little bit about Glympse science, because I think right now it's a totally innovative technology, and I'm very excited about what this is going to do in a number of indications, but particularly for oncology. So at Glympse, we engineer synthetic biomarker precursors that we can then inject into a patient. They then travel to the tumor, and they're processed by specific enzymes of interest, like in the tumor microenvironment, releasing barcodes that are then read out of a simple urine test after an hour. So this allows us to monitor real-time processes like tumor cell growth, drug response. For example, in immuno-oncology, we can measure granzyme B activity when activated T cells engage with tumor cells. Or in any mechanism of action for drug response, we can look at caspase activity as cell death occurs in response to therapy. Because these are engineered sensors, we're not reliant on a naturally occurring synthetic or circulating biomarker. We can customize these to measure biology of interest, even where such a biomarker doesn't exist. And we can tune it to achieve the required sensitivity that would be necessary. And then because these are non-invasive, we can look longitudinally at both the tumor and the microenvironment over time without having to perform a biopsy. So I think the innovation that this technology is going to be able to provide will be to partner with drug developers to get an earlier look at how their compound is performing in people. To get at a week or even 2 weeks after therapy is administered, to be able to see in real time the changes that are occurring and to understand better sort of the proof of mechanism for your molecule.
Do you think any of your readouts could have ultimately become sort of registration markers as well, or is it going to be for sort of early signals?
For oncology, right now we're thinking that this first product is going to be more of a translational marker, but should it actually be demonstrated to show that you can predict response, or perhaps more importantly in this case, non-response, this could be something that we would take forward to registration that would allow, say, a community oncologist to be able to measure whether a patient is responding to their current line of therapy, and if not, get an early decision point to then move them on to something else.
Great, thank you. So moving on to you, Rick. So you're sort of sitting on the front line in terms of those early clinical studies. I mean, we've all seen, the huge impact that the immune checkpoint antibodies have made to, this sort of the treatment options for a number of different patients. What are you seeing that's looking sort of promising on the horizon in terms of sort of early clinical signals, and what are you excited to see readouts in the next few months?
Yeah, so I think for us, it's really about, trying to think about combinations and, really being intelligent about the combinations and, being, even, and combinations that are maybe even beyond PD-1. sorry, Emma, I know you're with BMS, but what combinations do we have that are beyond, that are really beyond, what PD-1s are? I think where we have to look at this is starting to think about, we know that, despite the, tremendous success of PD-1s and anti-PD-L1s, and even to some degree CTLA-4s, that they're just not enough. So, part of the combinations that we are thinking about is, do you actually need to hit the cancer immunity cycle in multiple spots to sort of, ramp it up, and increase antigen presentation, get rid of M2 macrophages. So we are thinking about, those other ways to sort of complement and not necessarily compete against PD-1s, but things that actually add to it. But we're also thinking about other things like, do we think about combinations with, standard care chemotherapies or radiation, things that are routinely used in the clinic that could help, really start immunogenic cell death. And really start thinking about, what are those innovative combinations that are based upon the biology that's being driven. And I think certainly the other thing, and I think Wendy touched on this, is, certainly I'm looking at our clinical trials and, just the enormity of data that we get out of our clinical trials in IO. And I think it's really, part of it is because we have to kind of like throw, and I know Emma, you mentioned this a few days ago as well, we really throw the book at these things to try to, really just detangle the complexities of the immune response that we're seeing. So I think part of it for us is really, working with companies like Wendy, like Glympse, to actually see, are there ways that we can earlier and earlier, get a read on these, on our drugs that are working. So I think, those are the things that we're really interested in. And for those of you that just saw the ACR, it seems to us that, the TIGIT trial that just showed some pretty nice results, they went from an escalation experiment that, that really didn't show all that much. They must have seen something in their translational data that really focused their, this is Genentech's trial, but it really focused their later trials to really help them see and then get a nice response when they went into combinations. So, I think, we are starting to realize, sort of this translational medicine that everybody kind of throws around. So, that's, I'm excited to see, this really move into the clinic and, help us understand what's going on.
Great, thanks. And Stefan, so I mean, you're looking at sort of new stuff all the time. So, kind of what's really exciting in from your vantage point at the moment?
Yeah, we really see many very different approaches at the moment in oncology. And I mean, I was thinking about what is really innovation, and initially innovation is like a new idea or a creative thought or something like that. But I think it needs to translate into something meaningful for a patient, right? Innovation itself, as long as it doesn't translate, is maybe a little bit meaningless. But when I look at all the ongoing trials and at the landscape, actually, I find a couple of things quite interesting. So, for example, what we see is in the heme cancer space, there is quite some success for T-cell engagers, like direct T-cell engagers for multiple myeloma, for B-cell malignancies. I think these data are quite spectacular, I have to say. Then also cellular therapies. I think cellular therapies are getting more and more momentum. And initially, it was more the typical CAR T cell therapies. But now we see more and more that also include neoantigens, very personalized therapies that have a completely novel way of dealing with cancer and making a therapy, right, for each individual patient. It sounds weird, but actually, cancer is not a simple disease. So it's a very personal disease. So actually, it makes sense that it needs a personalized treatment to some point. Then I think we shouldn't forget about all the oncogenic drivers. So there are still plenty of oncogenic drivers. The markets may get smaller and smaller. and we may get more and more into this resistance mechanisms like EGF receptor, third, fourth generation. I think there's a huge potential if we can handle somehow the commercial opportunities. Then for me, surprising are also the antibody drug conjugates. There are quite amazing data in the HER2 field, for example, Daiichi's compound. I think this is amazing to see how much can be done in this field. And I think we mentioned before myeloid cells. At the moment, everybody, or when it started cancer immunotherapy, everybody focused on T cells because they are the typical killers. But it's much more complicated, right? Dendritic cells, macrophages, M2, M1 macrophages. I think there's much more to gain on this. And finally, my personal favorite is still early diagnosis. So you know, whenever
see Wendy waving the flag.
Yeah, because you know, in the end, whenever I can see a cancer patient fully metastatic and a heterogeneous disease everywhere in the body, I mean, what treatment, one-shot treatment should cure this patient or make it chronic, the disease? I can see my personal vision is, and also the vision of Roche is a little bit to move earlier and have less harsh therapies and detect the cancer earlier, and maybe do something even like a screening. You know, you can maybe screen the population, and maybe the current methods like colonoscopy and others are not so convenient. But if we would find something like a blood-based test, we could get at least a hint this patient or this person could have cancer, and then you know the organ, then you do a more detailed diagnostics. I think we could catch it much earlier. And maybe surgery would be already enough to treat many patients. So maybe we would not even need this very unkind treatments. And honestly, when we had our, we always do an oncology summit at Roche each year. And when we had a patient last year, I mean, for me, it resonated a lot with me when he talked about his disease, that he was in particular interested in getting kinder medicines. And you know, not that the medicine itself almost killed him. In the end, he was a cancer survivor. He was lucky, but I think that could be the path in the long term. So well, I think that is interesting areas.
No, well, I think that, I mean, it's easy kind of as a, you know, as a drug, develop. I mean, you fall in love with your medicines, what you're trying to do, but you realize that the cancer patient is still trying to conduct, you know, their life, right? They're still trying to go to work. They still need a standard of care. And I remember, you know, a chemistry colleague once who, you know, then got diagnosed and went on to sort of chemo, and you know, and as a chemist, he said, we have to make more selective compounds. Yes. He said, like, this collateral damage is just, it's just, you can't tolerate it, things like that.
Absolutely, Emma. And I think also, unfortunately, from some personal, relationships we all have probably with cancer patients, we know that we shouldn't say, well, it's a cancer patient, those patients, they have nothing to lose, it's their life, they can maybe accept more side effects. No, that's not the case. This is usually a patient that is in his last month or years of life. I think also these patients, they have a right to have a good treatment and a treatment that is somewhat acceptable to them, and that also increases the quality of life and not only the length of the life. So that is one of the most pressing areas for me, early diagnosis. Like in cardiovascular disease, there's also more and more going into prevention, into early diagnosis. So that is more or less my
right. No, thank you for that. No, I think, and perhaps sort of, Wendy can sort of build off that in terms of your vision. And I sort of want to bring it back as you sort of comment on that, Wendy, in terms of, we talked about, like, it's amazing that we haven't really kind of lost pace in the pandemic in terms of what we're trying to do and where we're trying to take oncology. But, my guess is that we're still sort of building off a head of steam that was started kind of before the pandemic, and sort of, so, I mean, do you have any fears about sort of long-term consequences? I mean, again, with early diagnosis and things like that, again, as we're looking at biomarkers, what's the implications for underlying viral infections in patients that affects then kind of what are we reading out, or ability to sample patients? We're going to be in more of an era of telemedicine and things like that. How's this going to complicate kind of what we're trying to do in the early diagnosis field? And Wendy, I know you've lived and breathed this for a long time, so perhaps I'd love to get your opinion on that.
Sure. I mean, yes. So early detection, I think, for all the reasons that Stefan said, is really indeed the holy grail that we're all shooting for. If we can find a patient who has a stage 1 tumor and it's localized disease, we can remove it with sort of a minimum amount of morbidities associated and probably affect a cure. I think the advent we've seen with multiple companies that have come through with circulating tumor DNA sequence is really exciting. We know that it's possible to detect and profile tumor-specific mutations from plasma, but it's still really challenging to do that in some indications, particularly for the stage 1 tumors. I'm encouraged by the progress, but there's still more that needs to be done, and we need to bring the costs of those tests down. You can get the sensitivity you need from these tests with a $10,000 sequencing run, but that's not something you can use every year. Like you would need to if this becomes a routine screening test. But I do.
Carry on, sorry.
Oh, no, that was basically what I was going to say.
Oh, no, I was going to ask, and it's probably asking on Rick's behalf, but really on all of our behalf. So, I mean, again, we are developing all of these different readouts to sort of try and monitor what's going on. Right now, kind of the number of options of readouts is continually expanding. And because we don't really know which ones are truly indicative or kind of, everyone is measuring everything. When do you think we're going to start getting back down to figuring out which ones are most meaningful? What's your sense based on what you've seen?
So I think, when I was at Novartis, we were like throwing the book at it, right? Yeah. We were trying everything we could. Some of that was guided by biology. Some of that was guided by what was readily available in our hands. So I do think that we're going to learn more from both, especially in early trials, by doing this type of hypothesis-free exploration with broad tests like RNA sequencing or other proteomic type approaches where we can really understand the biology that we're seeking for. But then I think going forward, it's going to be a test that can get us to the core biology that it is we're trying to understand, right? A lot of the measures that we had at hand as we were exploring, checkpoint inhibitors and combinations were somewhat opportunistic, but they weren't real-time measures. They were, months distant in time where we were trying to predict off of a biopsy well before a patient went on to therapy. And some of these things aren't going to be predictive. We're going to need to actually measure what's occurring in the tumor. So the more we have longitudinal monitoring types of tests, such as ctDNA sequencing for things like measuring EGFR mutations, or what Glympse is building, where we can have a non-invasive measure of tumor biology going forward, I think that will be really help us to better understand.
And Rick, so in terms of, so you're saying you guys are still, at the book throwing, in terms of testing out lots of different things. I think, again, not so, I think most of the industry is, but kind of, how are you trying to whittle down, to kind of which ones are most meaningful? Obviously, when we, don't have enough patient samples and the cost of doing, the cost of running trials with all of these different sort of readouts. What's your thought there in terms of how we get down to the meaningful readouts?
Yeah, no, so I agree with you. I mean, I think if we remember back to Jurassic Park, it's a question of just because we can do something doesn't mean that we should do it. If we remember that quote, but I think one way that we're trying very hard to do this is to sort of standardize things across Takeda, and we have many different assets that are using a CyTOF panel. Well, we don't want to be developing a CyTOF panel for each of the different assets that we have. So we're trying to make core CyTOF panels that we can apply. But then the true value of that also comes in, and I think everybody likes to use the word reverse translation. So since we're using the same panels, we can then sort of begin to interrogate our samples very prospectively from all of our studies. So we are trying to really start to build a consistency across the organization of what we're doing, so that each trial is not doing its own little its own thing. So we do want to make sure that there's that consistency and really focus down on that. And as both you and Emma and Wendy have noticed, we are looking at really breaking down our questions very simply from a translational perspective. Are we hitting our target? Are we hitting the biology that we need to understand? They're not hard questions, and we're really focusing in on those questions that we can, and then the rest of it is sort of exploratory, and we're actually trying to really predefine what success looks like. So really whittling it down into what are those key biomarkers that tell us that we're trying to do what we hope to do. And as Wendy said, some of it is practical, like TCR sequencing takes a long time, and you need X amount of samples. So that might not be our decision-making biomarker, although that really does enhance it does really enhance our understanding of the biology and sort of that adaptive immune response. So so I think for us it's about sort of prioritizing, and it's about sort of building this like cohesive translational aspects across all of oncology, and even through our GI diseases that we certainly interact with them, that there's a lot of inflammation diseases that we try to learn from each other. So it's maybe also look outside of oncology as well, because there's a lot for those of us in big companies, like try to avoid the silos.
Yeah, no, thank you for that. So we have a number of questions coming in from the audience. So I'm just scanning through to see kind of which ones we've covered and which ones to send to you guys. So there was a question, and Rick, I mean, I'll ask you this question, and I know certainly we've been discussing it at BMS as well in terms of coming back again to this sort of long-term sort of implications of this sort of the pause in terms of the potential implications to our pipelines. I don't know kind of what's happening at Takeda. I know certainly BMS shut down all their trials. We are getting them back up and running, but certainly we can't get everything up and running at the same time, so we're having to do it slowly. So there will be a sort of an impact in terms of being able to get drugs to our patients. I mean, certainly in terms of access of registered drugs, we've tried to, sort of make sure, we've had all of our manufacturing open during the whole pandemic. And, and then we've sort of, created, and I know a number of companies have been doing this as well, in terms of, trying to provide, either free drug or whatever to, patients that have lost their sort of healthcare. So there's a number of things that we're doing as a company to make sure that the impact of the pandemic does not impact, patients and their suffering. Perhaps, is there anything else you'd like to add to that, Rick, in terms of, as you ramp back up, your thought in terms of, will we see a scar in terms of our portfolio and the pipeline from the industry because of the shutdown?
I mean, to say that there's not going to be an impact is, I think, is not true. I mean, certainly we've all been slowed down, but I think we've also gotten very creative in working with vendors. I mean, similar to I think Wendy and Emma and Stefan, we've gotten creative with as we were shutting down, China was opening back up. So we have a lot of CROs that we work with in China, or CROs in Canada that we are working with. And I think we're also getting a little bit more creative, a little bit more creative in our early clinical trials. So instead of maybe just thinking about US, we are also thinking about opening our trials in somewhere in Southeast Asia where coronavirus is much better under control. So I think we are trying to be creative and be a little bit think outside the box a little bit for the ways that we are conducting ourselves. And certainly our late stage clinical trials weren't affected. Those that have a safe dose, those that like our later stage were still prioritized. Those still kept going. Like our phase 3s weren't really affected, and some of our really high profile phase 1s were even still going, that we knew we had a safe dose, we knew we had safety. And so sort of these first in human ones though were impacted because they are very immune stimulatory. And what we know about COVID is that it is sort of a CRS type reaction. So thinking about throwing an immune agonist onto CRS, do we really want to take that risk? And I think, that's something that we're thinking about. But looking forward, I think, yeah, the pipelines will slow down a little bit, but I think, what our labs have also been open, key projects have really forced us to prioritize, which sometimes isn't a bad thing either. So, sometimes we have to, I think sometimes prioritization is good.
Yeah, no, thank you for that. I'm going to just throw a very specific question to you, Stefan, and we probably maybe have a minute for you to answer this. So someone from the audience had a question about real world data and how we're using it. The question sort of referred specifically to Flatiron, but I'm sure you have access to other real world data as well. So perhaps you could talk about sort of the access to that and how that impacts kind of, how that helps preclinical research and clinical development, and how Roche is applying that.
Yeah, I mean, we have access to Flatiron, for example. We have access to FMI, which is the sequencing data. So I think altogether this real world data could in principle help to speed up drug development, because you can run like virtual trials, right? you know your patient population, you know the history, you know the treatment of this patient population. So I think overall this is now getting more and more implemented also in our trials. But I have to say, these are still, of course, early days. So we tap into this information for sure, also in our clinical development programs. But I think we are in the beginning still. So I think it's a little bit too early to share a deeper experience or expertise. But I think for us at Roche, it's a huge asset to have access to this real world data.
Excellent. Well, I just really want to thank all of you for the conversation today and sharing, different, your different perspectives in terms of, obviously, a huge area that sort of continues to sort of innovate, and also to get your sort of your viewpoint in terms of how the pandemic has impacted it. I want to sort of leave with sort of one sort of aspirational vision and maybe sort of commitment from some people around the table. I think what we've all been surprised by is really just how, as an industry, kind of how the force of it when you all really come together and work together. I mean, if you think about all the sort of real-time reporting of data as we've understood what's happened in the pandemic, and sharing of clinical data, and what we're learning from patients and antibodies and T-cell responses and stuff like that. I mean, if we could sort of apply that immediate learning to the question we were talking about earlier, right? Early diagnostics and stuff like that, rather than everyone doing it sort of in their isolated bit. If we could bring, one lesson in terms of those shared learnings and sort of, try and get our industry, talking across lines more often, I think, the power of what we all bring to the table is just absolutely incredible. And just the speed by which we can advance medicine is really huge. So with that, thank you, guys. Real pleasure to spend the time with you this afternoon, and I really want to thank the audience for listening in. And I hope that you found the conversation interesting and stimulating.
Thank you, Emma.
Thank you.
Thank you.
Thanks so much.